Go.
Okay, this is my slide deck pitch for the PCR lab that we did. The research question that we were trying to answer was, am I homozygous or heterozygous for the allele gene on chromosome 16? And the purpose and objective was to learn PCR and analyze data. So PCR was the technique that we used to do this lab, and it stands for polymerase chain reaction, and so it copies specific pieces of DNA over and over again in vitro, which basically just means in a lab. So the methods that we used on day one, we did DNA extraction, which was where we swished salt water in our mouths, and then we did a bunch of steps to extract the DNA from the salt water. On day two, we set up the PCR tubes, which had the master mix and the primer. On day three, we actually ran the gel, and on day four, we dyed it and we analyzed it. So the data that our group got, our data was invalid. So these are two pictures of it. So it shows right here the ladder, which was the positive control, which means that it should have worked, which it did. And then one of ours stayed in the well, but none of them actually ran. For the analysis, human errors, we may have not properly created the gel, or the teacher may have not properly created it. There could have been a hole somewhere that caused none of them to work. And issues with pipetting, because our class has a lot of trouble with pipetting. And a procedural error would be the wrong voltage on the gel box. So on one of the steps, it said to do 150 volts on the gel box, but we had to change it to 120 so that it would be correct. The limitations was not making our own gel because we didn't make it ourselves, and we weren't able to turn it off because of time constraints. And some practical applications for PCR is finding a genotype for specific genes, like we were trying to do in this experiment, and crime scenes, so trying to match blood and find blood type. So the conclusion, I don't know the answer to the research question because there was no data to find the answer, so I'm pretty confident that I know that it didn't work. And for future research, we need to figure out what caused the problem of none of them being able to run, and then solve it and repeat the experiment. Any questions?
Thanks, Sarah.